Good morning everyone and welcome to the botany lecture. Today's topic is classification of plants, and prior to the course Linnaeus, the botanists used about 20 words to name a plant, and that was not very convenient. So that is why the binomial system that was developed by the Carl Linnaeus was so popular and was adopted very fast. So the Carl Linnaeus introduced a binomial system, and that system is very useful and very convenient. And the binomial system gives two names for every plant. Like we have a first name and second name, or the given name and family name. The same is for the plants, and plants also have two names. So this is a very popular garden plant, spiderwort, and is also medicinal because it's ends for the wort. So prior to the Linnaeus, the botanists used Latin name for this, that is Trascantia ephemerum phalangides ripetalum non repens virginiatum gramineum. So this looks like a very complex name and hard to remember. So if we translate this name, that means the annual upright Tradescantia from Virginia with the grass-like habit, 3 petals and stamens with hairs like spider legs. And hence the name of the plant spiderworts, because it looks like a spider legs. And Linnaeus used this lengthy scientific description as well, but he added it the margin a simplified scientific name for this plant, Tradescantia virginiana. So thus Linnaeus' most significant contribution to biology was binomial nomenclature, a system of naming of plants based on a unique two-part name for each. The first part of the name designates the genus, and the addition of the second part, the scientific epithet, designates the species. The specific epithet is usually a word that describes some particular quality of the organism. Like we have a family name, the plants like my family name is Kamaliv, and my given name that is specific to me is Rafis. So my full name is Kamaliv Rafis. The same is for the plant. The plants have the genus name. In the case of the Tradescantia, it is Tradescantia. So this is Tradescantia, and the specific epithet is Virginiata, and Virginiata is specific for this species. The generic name can be used alone to describe all species in the genus, but the specific epithet is never used alone. It is always preceded by the full or abbreviated generic name. In each specific name, the generic name is first, and its first letter is always capitalized. Like Tradescantia is always written from the capital, and Virginiana is a specific epithet, and it's written from the common letter. So the specific epithet is not capitalized. Both names must be underlined or italicized. Like here, the font is italic. Corn, for example, is in the genus Zea. Its specific epithet is maize. The proper scientific name for corn is Zea maize. So when you eat something from the corn, it comes from Zea maize. Also, any specific epithet is used for only one species within each genus. It can be used in more than one genus. For example, Quercus alba is the scientific name for the white oak, and no other oak species can have alba as its specific epithet. However, Nymphaea alba is the scientific name for the European white water lily, and Salix alba for the white willow. So alba is actually translated from the Latin as a white. Both parts of an organism's scientific name must be used to accurately identify the species. Scientific names are generally composed from the Greek or Latin roots, or from Latinized versions of the names of persons, places, or characteristics. Hedda, the generic name for the ivy Hedera canarensis, for example, is based on the Latin word for ivy. The specific epithet canarensis tells us that this plant is native to the Canary Islands. The use of Latin, a dead language that does not change, rather than a modern language in naming organisms, is a carryover from the days when Latin was the language of European scholars. Why do we continue to use Latin rather than common names for plants and animals? Why call a sugar maple Acer saccharum? Acer is derived from the Latin word meaning maple, and saccharium from the Greek for sugar. The main reason is to be precise and to avoid confusion, for in some plants of the United States, this same tree is called the hard maple or rock maple. The plant Mirabilis jalapa is generally called 4 o'clock, but some people call it marvel of Peru, and others call it beauty of the night. So to my mind, beauty of the night is a very beautiful name. And uh, uh, because of the many instances of confusing common names, exact scientific names are important for accurate identification of organisms. In addition, many scientifically important organisms lack common names. Since each organism has only one scientific name, research in Canada reading a study published by the German scientists knows exactly which organisms were used. And same is for the like uh, Egyptian students that reads about uh, mm, the uh, the from the textbook that is published in Russia, for example, uh, also will understand the name of the plant and will identify plant correctly. Sorry. A complete scientific name has a third part that cites the person who formally described the organism. Thus, the complete scientific name for the spiderwort is Tradescansa virginiana Linnaeus. Uh, and usually the name of the person is not entirely precise. The author's citation is often abbreviated as in Tradescansa virginiana L. The draft index for author abbreviations and other sources explain the abbreviated names of author citations. The author citation is useful because it helps researchers find the original description of a particular part. The species is the basic unit of classification. I think you remember from the previous lectures that species consist of one or more population whose members can interbreed to produce fertile offspring but are reproductively isolated from the other organisms. Members of a species share a common evolutionary ancestry. The species is the basic unit of classification, but not the smallest taxonomic group in use. Geographically distinct populations within a species often display certain characteristics that distinguish them from other populations of the same species. If they can interbreed, however, they are not truly separate species but are known as subspecies. Experts can usually distinguish subspecies from one another. However, species may grade into one another at the borders of the geographic ranges where there is opportunity to interbreed. Some of these subspecies are apparently in the process of becoming reproductively isolated, and many over time become separate species. Thus, some subspecies provide biologists with the opportunity to study evolution in progress. Plants that are produced using techniques such as selective breeding, also called artificial selection, are called cultivars. The term cultivar is an abbreviated form of cultivated variety. A cultivar is not equivalent to a subspecies. A cultivar is produced while under cultivation, whereas a subspecies is geographically distinct population that evolves by natural selection. Cultivar names may be used after scientific names. For example, two cultivars of peaches are Prunus persica cultivar rosea and Prunus persica cultivar early flame, which is a nectarine, uh, and nectarine is a smooth-skinned cultivar of peaches. Peaches are very popular fruits, so nectarines are the cultivars of peaches. Uh, Classification is hierarchical. The narrowest category in the Linnaean system is the species. So the species here is the most narrow, and the broadest is the kingdom. Kingdom is plant for the plants. Closely related species are grouped in the next level of classification, the genus, and the genus are like after the species. One or more related genus are assigned to the same family. Like we have a family, the plants also have a family, and families are grouped into orders. Orders into the classes, and classes into phyla, and phyla into kingdoms. And kingdoms are grouped into domains. There are 6 domains. These groupings can also be separated into subgroupings, for example, subphylum or subclass. Each taxonomic level is broader than the level below. For example, the family Zingiberaceae, the ginger family, contains about 1,300 species in 14 genera, including genus Zingiber, the ginger genus, genus Curcuma, the turmeric genus. Well, by the way, do you have a curcuma in Egypt? Asma? So it looks like you are not wake up yet. So the genus Eletaria, that is a cardamoms genus, and the family Zingiberaceae, along with 4 other families, is placed in the order Zingiberales. Order Zingiberales and 18 other orders belong to the class Monocotyledons, that are monocots, one of 2 main classes of flowering plants. Like you had classes in school, the same is here. The plants also have classes. The monocots produce seeds that contain 1 cotyledon and possess flower pods in 3s or multiple of 3. Class monocotyledons and class eudicotyledons, along with the several minor classes, are grouped into phylum Anthrophyta. So the monocotyledons and eucotyledons grouped to the Anthophyta and Anthophyta, which along with the phylum Coniferophyta. Coniferophyta are actually conifers that are Christmas trees. So it's Christmas very soon, so it's a very important tree. and phylum Gnetophyta and several others belong to kingdom Plantae. So Plantae are the kingdom that is most important kingdom in the world. So are actually plants. Most taxonomies identify organisms primarily on the basis of structural features. Using such features, taxonomies have developed special guides known as dichotomous keys to aid in identification of unknown plants. A dichotomous key consists of a series of two contrasting statements made about plants. The person trying to identify the unknown plant chooses which of two statements is correct for the plant in question and is then directed to another set of paired contrasting statements. By working through the statements in the key, the investigator eventually identifies and classifies the unknown organism. To understand how such a key works, let's try to identify the plant in on this picture. Sorry, on this picture. The uh, the entry in the right hand column opposite the appropriate structural features identifies the plant or tells you which number to go to next. So be aware that students often have difficulty using such keys because they are unfamiliar with the terminology or because they make a wrong turn in haste. So the classification of plant and other organisms into groups determined by the evolutionary relationship is called systematics. A systematic search for the evolutionary history of phylogeny once these relationships are defined, the classification of organisms is based on common ancestry. If all the plants within a taxonomy group share the same common ancestor, the group is referred to as monophyletic or one branch. Monophyletic groups are natural groupings as they represent true evolutionary relationships and they include all close relatives. Many groups are polyphyletic, consisting of several evolutionary lines and not including a common ancestor. Polyphyletic groups may misrepresent evolutionary relationships. For this reason, taxonomists try to avoid constructing polyphyletic groups. An approach to systematic that has been widely adopted in the past several decades is cladistics. Cladistics emphasizes phylogeny by focusing on when evolutionary lineages the lines of descent and they are divided into two branches. Cladist taxonomists who practice cladistics insist that taxa be monophyletic and use carefully defined objective criteria to determine branch points. Thus, common ancestry is the basis for classification. A cladist would classify diazids with flowering plants rather than with ferns because daisies and other flowering plants share a more common recent ancestor than do daisies and ferns. Cladists develop branching diagrams called cladograms. So these are cladograms. For example, daisy is actually more like flowering plants because it has flowers, or rather than the ferns. So the general goal of cladistics is to reconstruct phylogenesis using an analysis of evolutionary changes in specific characters. Structural, physiological, and molecular characters can be used for the analysis. The characters must be homologous, that are identical by common descent, and must have evolved independently of one another. The first step in constructing a cladogram is to select the groups. Here we use 4 plant groups: moss, fern, pine, and daisy. The next step is to select the homologous characters to be analyzed. In our example, we use 3 characters: vascular tissues, seeds, and flowers. For each character, we must define all of the conditions. For simplicity, we will consider our characters to have only 2 states: present and absent. The last step in preparing the data is to organize the character states into the correct evolutionary order. The most common method of doing this is by out-group analysis. An out-group is a group that is considered to have diverged earlier than the other group under investigation, and thus may represent an approximation of the ancestral condition. In our example, moss is the chosen outgroup, and its character state are considered ancestral. Therefore, the character state absent is the absent, so moss don't have vascular tissues, don't have seeds, and don't have plants. The state absent is the ancestral condition, and the character state present is the derived condition for each of the characters. If we examine the table and notice that all groups except the outgroup share a derived trait, vascular tissues, like this vascular tissues present in ferns, pines, and daisy, and no vascular tissues in moss. We may therefore conclude that these 3 groups, fern, pine, and daisy, are monophyletic, and using these data we may construct a preliminary cladogram. So this cladogram is the preliminary. The branch points as nodes represent the divergence of the ancestral lineage into 2 lineages. And thus node 1 represents the common plant ancestor there, as well as the divergence of the outgroup moss. So this is common ancestor and divergence to mosses, and the common ancestor of the 3 vascular plant groups. Similarly, node 2 represents the subsequent divergence of the fern and the ancestor of plants with seeds. So node 2, the branching process is continued with the data from the another table, Note that pine and daisy are more similar to one another like this because uh, than to any other groups. The presence of a common ancestor at node 3 indicates this relationship. The distance from the base of the tree indicates the relative time of divergence. For example, the farther a node is up the tree, the more recent the time of divergence. In our example, node 3 represents the most recent divergence and node 1 represents the most ancient divergence. Thus, the daisy is more closely related to the pine and more distantly related to the fern. Until relatively recently, the broadest level of taxonomy classification was the kingdom. The history of taxonomy at the kingdom level is an excellent example of how scientific knowledge expands and changes over time. From the time of Aristotle to the mid-19th century, biologists divided the living world into two kingdoms, plants and animals. With the development of microscopes, it became increasingly obvious that many organisms could not easily be assigned to either the plant or the animal kingdom. For example, the unicellular organism Euglena, which has been classified at various times in the plant kingdom and the animal kingdom, carries on photosynthesis in the light but in the dark uses its flagellum to move about in search of food. So, like uh, this one, flagellum. So, more than a century ago, a German biologist, Ernst Haeckel, suggested that a third kingdom be established, the kingdom Protista. So, um, simple and ambitious organisms such as bacteria and most microorganisms, including uh, kingdom in kingdom Protista. Thus, protists are a diverse group of mainly unicellular, mainly aquatic organisms, and they are very important in medicine because most of the diseases are caused by the bacteria. In the 13th, uh, the French biologist Eudore Charton suggested the term prokaryotic before nucleus to describe bacteria and the term eukaryotic uh, true nucleus to describe all other cells. Virtually all biologists now accept the separation between prokaryotes and eukaryotes as a fundamental evolutionary divergence. In 60s, advances in electron microscopy and biochemical techniques revealed basic cellular differences that inspired many new proposals for classifying organisms. In 1969, Whittaker proposed a five-kingdom classification: Monera, Protista, Planta, Fungi, and Animalia. Uh, based mainly on cell structure and the ways that organisms obtain nutrition from their environment. Kingdom Monera was established to uh, accommodate the bacteria, which are are fundamentally different from all other organisms in that they are prokaryotic and lack distinct nuclei and other membranous organelles. Uh, Whitaker also suggested that fungi, such as mushrooms, molds, and yeast, be classified as a separate kingdom fungi, rather than as part of the plant kingdom, because fungi are non-photosynthetic and obtain energy by absorbing nutrition. Fungi also differ from plants in the composition of their cell walls, in body structure, and in reproduction. Most biologists accept the five kingdoms proposed by Whitaker until DNA studies by Carl Woese and other beginning in the late 70s revealed that there are two groups of prokaryotes. Woese proposed that the prokaryotes account for two of the three major branches of organisms. Whose hypothesis continued to gain supporting evidence, and now most biologists think the two groups of prokaryotes, the more familiar bacteria and the distinctive archaea, are each merit their own kingdom. So archaea is actually the sixth kingdom. As a result, many biologists currently recognize a sixth kingdom scheme that makes into account these evolutionary relationships as well as cell structure and ways of obtaining energy. The six kingdoms are bacteria, archaea, Protista, fungi, planta, and animalia. Most biologists are also use a level of classification above the kingdom called a domain based on fundamental molecular differences among the bacteria, archaea, and eukaryotes. They classify organisms in three domains: bacteria, which corresponds to the kingdom bacteria; archaea, which corresponds to the kingdom archaea; and the eukarya that are actually all other kingdoms. Biologists have inferred that the three domains are the three main branches of the tree of life. Also, you might think that there are two domains of prokaryotic organisms would be closely related. The domain archaea is apparently more closely related to the domain eukarya than it is to the domain bacteria. Protists do not fit well in a single kingdom. Biologists use molecular data and ultrastructure to clarify evolutionary relationships among the protists. For example, biologists compare the nucleotide sequence for the same gene found in different protists. Ultrastructure consists of the fine details of cell structure reviewed by electron microscopy. Both molecular data and ultrastructural suggest that members of kingdom Protista are not a monophyletic group. Instead, the most biologists regard the Protist kingdoms as a paraphyletic group. The common eukaryote ancestor that gave rise to the members of kingdom Protista also gave rise to plants, fungi, and animals, all of which are multicellular eukaryotes not classified in the Protist kingdom. Some biologists want to resolve this problem by splitting the organisms in the Protist kingdom into several smaller kingdoms, each with its own common ancestor. However, most biologists, including us, would rather deal with the limitations of a single monophyletic Protist kingdom than with several additional monophyletic kingdoms, because we have to learn more kingdoms. That is not very convenient. And as you have seen, scientific knowledge changes as new information comes to light. It may be that Kingdom Protista, despite its convenience, will be partitioned into several kingdoms in the future. We say more about the organisms in the Protist kingdom later. So in next semester actually. So that is all for today, and thank you for attending the last lecture. Uh, and actually, if you have any questions, and we will continue in the next semester in February. So good luck with your exams, and see you next year.